Hi, my name is Jessica Hansen, and it is May 21st, 2022. I'm going to be reviewing the Unit 4 case reviews. I will be doing Case 1 and Case 2.1. First, we can do Case 1. Asked me first to discuss the normal mechanisms in which amino acids are broken down. Free amino acids are in the liver and the muscles, and they'll undergo a reaction called a transamination reaction. Transamination is when the amino acid, let's say alanine in this case, is interacted with alpha-ketoglutarate. This interaction uses alanase aminotransferase, which is abbreviated ALT, as well as a PLP cofactor to make a new keto acid and a new amino acid. The new keto acid would be pyruvate, and the new amino acid would be glutamate. Glutamate is then shuttled to the liver where it can be acted upon by glutamate dehydrogenase. This reaction includes introducing water and NADP+. The results of this reaction are alpha-ketoglutarate, NADPH, and NH3, which is ammonia. This glutamate can be acted upon by glutamine synthetase within the astrocytes of the brain, which can help take up the ammonia from tissues and create glutamine. Should glutamine, however, remain in the brain, it can be dangerous because it is osmotically active. This will encourage water or draw water into the brain or into the cells and can create cerebral edema or edema within other cells as well. Left unchecked, this edema can lead to a coma or brain herniation or even death. Glutamine that's returned to the liver will be acted upon by glutamase, which will free the ammonium and create ammonium and glutamate again. This ammonium, as well as the ammonia and ammonium that were created from the initial interaction with glutamate dehydrogenase, will then enter the urea cycle. Also asked to describe how nitrogen removal is connected to gluconeogenesis in the fasted state. During the fasted state, the body creates endogenous amino acids that can be used for gluconeogenesis. Like I mentioned before, when glutamate interacts with glutamate dehydrogenase, it creates ammonia, NADPH, and alpha-ketoglutarate. This alpha-ketoglutarate can enter the Krebs cycle as a substrate and create substrates that will eventually be used in gluconeogenesis. And as listed above, when the alanine is going through its transaminase reaction, it creates pyruvate, which can also be used as a substrate for gluconeogenesis. In the case of this patient, I initially had a very clear diagnosis that it was ornithine transcarboxyl ornithine transcarbamylase, or OTC, deficiency. However, in doing research within scientific journals and comparing the provided lab values to the lab values in the journals, I also have a differential diagnosis of a possibility of deficiency in arginase. I will explain these. I would say that OTC deficiency is the most likely deficiency within this child. I say this because the patient's high ammonia level clearly signifies that there's a defect in the urea cycle. The high levels of urine orotic acid are most likely due to the backup of carbamoyl phosphate. The carbamoyl phosphate can exit the mitochondria, where it would eventually create orotate that gets excreted by the kidneys into the urine as orotic acid. There's further proof of the defect in OTC due to the patient having low citrulline levels. This shows that the carbamoyl phosphate is not combining with ornithine via the OTC enzyme to create an adequate amount of citrulline. If this is in fact an OTC deficiency, arginine, which is usually a non-essential amino acid, will become an essential amino acid, which means it now must be ingested by the patient. Arginine is generally found in high protein foods such as red meat, fish, However, this patient's going to have to be on a very low protein diet and very restricted with those kinds of foods, so may just need to take a supplement in order to get the arginine. The reason I find myself questioning this diagnosis is when I was reading through research studies, OTC deficiency in neonates is generally only found in males. Very rarely does it present itself in females, which I know doesn't mean it's not possible, but just what I was researching. It's also noted to create a very low citrulline level. It's stated in the single digits. This patient being a female and the citrulline level still being in the double digit, it led me to research into an arginine deficiency. This patient doesn't fit that deficiency perfectly either. Arginine deficiencies very rarely have as high of ammonia levels as this child has, and very rarely has this high of orotic acid levels as well. So based on the knowledge that I have from researching this, I would still assume an OTC deficiency. However, I would retain some reasonable suspicion for an arginine deficiency. In looking at this patient's lab values, we do know for certain, however, that the CPS1 enzyme, or the carbamoyl phosphate synthetase type 1 enzyme, is functioning correctly. We do know this because regardless of which enzyme deficiency she has, the patient is clearly producing enough carbamoyl phosphate, and that is something that is backing up within the system. Next, I'm going to review case 2, number 1. In this case, I believe the most likely scenario would be that the patient is deficient in branched-chain alpha-ketodehydrogenase, or BCKD. When there is a deficiency of BCKD, the branched-chain amino acids, which are valine, isoleucine, and leucine, along with their ketoacid substrates, which are alpha-ketoisovalerate, alpha-ketomethylvalerate, and alpha-ketoisocaproate, back up within the patient's system. There are no amino acids that become essential because of this deficiency. However, this backup and branched chain amino acids will inhibit the uptake of large amino acids, such as tyrosine and tryptophan. Tyrosine and tryptophan are used to synthesize dopamine and serotonin, which are very important. It can also inhibit the Krebs and ATP production, which can cause very high levels, because of the high levels of alpha-ketoisocaproate. This encourages edema via inhibition of the sodium-potassium pumps, and it also can impair cell-to-cell communication by inhibition of GABA synthesis. Having a deficiency of BCKD is a disease otherwise known as maple syrup urine disease. It was named this as patients with the classic disorder are noted to have the odor of maple syrup in their earwax, their sweat, and their urine. There are 5 types of the maple syrup urine disease, with classic, intermittent, and intermediate being the most common ones. Classic maple syrup urine disease is the most severe disease. It generally presents in the first 48 hours after birth, and this means that they have less than 3% activity function of the BCKD enzyme. If it's not treated in 7 to 10 days, there's an increased risk of death due to seizures, apnea, and cerebral edema. Intermittent MSUD is usually noted to have 5 to 8% of enzyme activity. It is the second most common type of this disease. It's generally diagnosed a little later in life and has irregular intervals of crises. The patients tend to have normal growth, normal development, and just have occasional episodes of catabolic stress. During these times of stress, though, the patients can have recurrent ear infections, ataxia, lethargy, seizures, and of course comas if it gets too severe. Intermediate maple syrup urine disease is said to be the least severe. It is when the mutation is in the E1 alpha subunit of the BCKD enzyme. Patients generally have between 3 and 30% function of their enzyme. Onset is noted at a later age, and it's generally correlated with how much function. So if they have more function of their enzyme, it'll be diagnosed at a later age. These patients can have acute neurological symptoms, irritability, dystonia, developmental delays, and seizures. Due to this particular patient's lack of symptoms and the fact that it was only brought forward because of her newborn screening and then she was readmitted to the hospital, I suspect the patient either has intermittent or intermediate maple syrup urine disease. The patient will likely undergo further testing. They'll have the levels of their branched chain amino acids as well as the keto acids, alpha ketone levels drawn, and genetic testing. I suspect the parents will also get genetic testing. The maple syrup urine disease is an autosomal recessive inherited gene, which means that both parents will have had to pass the gene on to this child. In the case of the older siblings being healthy and not having any symptoms, they should probably be retested. They may have something like the intermediate version where it was maybe missed on their newborn screening. In the future, this patient is going to have multiple dietary restrictions, including highly restricted protein intake. It's due to the fact that branched chain amino acids are in a myriad of foods. They're in eggs, most meats, milk, soy, nuts, beans. The patient's going to have a lifetime of blood draws. They're going to have to keep a very close eye on the plasma amino acid levels. And crises can easily be brought on by infection, stress, injury, or even fasting for too long. There are more aggressive treatments such as dialysis, and the only cure for it is a liver transplant. However, these are only generally seen in patients that have the classic version, and they are very rare. And a liver transplant will cure the disease, but it will not stop the patient from passing it on to their own children. So, thank you and have a great night.